I feel a little bit, how to say, it's very indynamic what I'm presenting here. Nevertheless, it has to do with magnetic fields, with electrons, and the application we use it for is also very dynamic, because the biggest application for electron microscopy in our days is in the end connectomics. How are neurons connected to each other? And what the electron microscopy adds to the field is how the structure actually looks like, which is so dynamic. So in this sense, there is probably also a need for high resolution information. And what I'm going to do is to talk a little bit about the basic contrast mechanisms, and then about a few different fields of application, how 3D information can be obtained with an electron microscope nowadays. And in the end, I also would like to talk about cryo applications, where we use unstained samples in electron microscopy, which is not possible in the normal applications. TEMs always have been used to shine through a thin lamella of prepared material, where SEMs have been used to show surface structures. These surface structures can be imaged at different length scales till very high resolution of the surface. And this is what long-time SEM and TEM have been separated because of the different applications. In nowadays, the SEMs, they can do also these kind of transmission images with a special setup between sample and beam pass. And in the SEM, in addition to this transmission mode, we have two other modes. We look at electrons coming back from a heavy metal element in the sample, so this can be detected, or we look at secondary electrons, which provides us surface information. So these are the two major different contrast mechanisms we have in the SEM. Secondary electrons are used to show surface structures, and where the backscatter electrons are used to show labeled membranes, to make labeled membranes visible, where you usually put an osmium on, and then we can see mitochondria and vesicles and endoplasmatic reticulum and all these kind of structures. So this is about my short technical or theoretical introduction to electron microscopy. A modern scanning electron microscope, they have modes for surface, for transmission, for 3D, and for ultra high resolution 3D structures as well. What is the background of these different contrast mechanisms? If the gray bar here is our sample, this one, then we have different detectors in the chamber of the system. One detector, these blue detectors here, are to look at secondary electrons. Then we have red detectors to look at reflected electrons, and we have another set of detectors for transmission. So the objective lens is always the same, the sample is always the same, little bit depends on, but the detectors are different, and with these different detectors we detect different properties of the sample. What I'm not talking about here is about elemental analysis. What kind of elements do these samples belong of? In this case, we look only at structures. Coming from the classical electron microscopy, which means transmission, what we usually have, we have a three-dimensional object. This has to be put in a hard plastic resin, has to be stained with heavy metals, and then it has to be sliced usually. And these slices have to be put on a substrate, on a carrier, sample carrier, and then they can be imaged. The classical way is this one here, and then these kind of carriers go into a TEM and can be imaged. This is but not only the TEM, you see there's a stem here as well, which stands for scanning transmission electron microscopy. If you look at this, how does it look in our days? We have these typical small grids with our samples on it. We have carriers like this 12 revolver carrier here. This goes into an SEM. The beam comes from the top, goes through the sample, then we have a detector underneath, and we can detect these different kind of samples. The nice thing is that if someone comes from classical scanning transmission microscopy then the sample preparation method has not been to change at all to take the same sample from a TEM to an SEM. The sample dimensions, the material labeling process is completely the same, and there's comparisons of how the same sample behaves in TEM, SEM, in transmission mode, and SEM in reflection. And as you see, at this level of resolution, we can see all the substructures of the kidney here in the same quality, where the stem mode maybe gives still a little bit better contrast. There are transmission electrons microscope TEMs available with a much higher resolution in the unstrained range, but these systems are not used for room temperature but for cryo. This is a different type of application. Here we talk about molecular configurations, but for structures, these systems all do the same job in the end. A few examples: muscle cells with the typical structures of muscles. Here the mitochondria. We can see this is vesicles of a neuron, and these structures are all easily to myelin surrounded nerve cells in this case, all easily to be detected with SEMs. If it comes to viruses, it becomes a bit more complicated. We can see viruses of different shapes here, but not these images we know from the TV that you can see the spike individual molecules. This is not the resolution what we achieve with the TEM, but we can see, count, and locate the viruses in this case. If you are interested in what can be done with SEM in terms of two-dimensional, there's a huge, nice database by Ben Diekmans. It's called Anatomy.org. So all kinds of data which have been acquired with TEM and SEM in 2D dimensions are listed in this database. And if it was a Zeiss image, then you can see there's a Zeiss logo, but also by a special browser which is available in the on the web page, which can be used to zoom in to these images, to show all the data of acquisition, and to share images and annotations with your collaboration partners. Nevertheless, what I have talked so far is that the same sample, these two classical TEM samples, can be used in TEM and SEM. There are some other applications where an SEM offers more options compared to a TEM. And this one is, for instance, that you can use larger sample carriers, larger samples, if you use an SEM. In this case, we do not care about the signal which is transmitted through the sample, but we look at the reflection of more bulky samples, which not necessarily has to be as thin that the electron beam can shine through the sample. This is nicely shown here in a work by Dr. Mike Reichert. What he has done is he used kind of a correlation approach where an SEM image was overlaid with the fluorescent image, not exactly of the same sample, but they cut rather thick samples of a micrometer. Normally, TEM samples have to be in a nanometer range, 70-50 nanometer. This is a micrometer sample, but here one surface of this cut was used for SEM, and the other surface was used for fluorescence labeling. And then you just put both images together, and you have a very simple correlation of the same surface by two different microscopy techniques. For electron microscopy, these samples are extremely large. It's a millimeter scale. They are 7 to 5 millimeters long. This is extremely big for electron microscopy, but it offers the opportunity to look at the tissue organization on the cellular level and the subcellular level as well, from different types of tissue, like here in kidney, brain tissue, and this was. All these techniques, what I have shown so far, is just simple 2D, smaller 2D, larger 2D areas. But if you take serial sections of your sample, then you can also assemble your individual 2D images to a 3D reconstruction. And there are three major technologies in SEM. This is array tomography, where you cut slice after slice, or line these slices and image each slice. That you can also do serial block phase imaging, where you have an ultramicrotome inside your electron microscope. You also get, in this case, you get surface images, which you align to a stack. And a similar technique is that you don't have an ultramicrotome, but an ion beam, which is used to take the sample material off and create new surfaces. I would like to introduce this ultrasound tomography. Is the simplest technique, so to say. And what you do here, you have your sample. You have an automated ultramicrotome in this case. Here, the individual sections are taken out by tape, so to say, and then these individual tape sections are aligned on a wafer, imaged by a special faster machine, because time is always the limiting factor here in this case, and then this can be assembled to a 3D sample in the end. What you see here is the activity of this ultra microtome, which is automated. This is the tape surface. This is the section which comes from the knife blade, so to say. Now it's taken up by this tape here, and this kind of operates totally automated. You can create something like 5,000 sections a day. Then you have a lot of material to image, which represents the 3D volume in the end. This tape has to be cut in sections. The sections have to be, the individual sections are aligned on the wafer. The wafer are stacked here, and then you have to image wafer after wafer. As you can imagine, this is a project which takes a lot of time, because we usually use only a single beam. And just to show you how such a, this is the wafer, these stripes of the tape. Here you see the individual sections. This is just a photograph from the mobile phone. At a certain point, we switch over to an overview image of the scanning electron microscope, which is somewhere at this point where you start to see the nuclei of the neurons. Now you see dendrites in this case. Somewhere the black stuff starts. The black stuff is the myelin. If you go further in, you see the mitochondria. Then we start to see membranes. Further on, here we see vesicles. So this is not only one image, it is a hierarchy imaging of 3 different resolutions. The number 4 one, the first one was the mobile phone. And if we do this not only one time, but from a complete stack of 2,000 images of a certain volume, then we end up with this kind of data set. So we have 2,000 images here representing this volume. And now we can start to track one neuron and see to which other structures this neuron is connected. This is what the connectomics application wants to do. They don't want to do this for 2 neurons only, they want to do this for the complete brain. And in this direction, what has been done, this was the first approach, but it shows exactly what's happening here. This is maybe 20 neurons out of these couple of hundreds. Continuing. I don't know. We should see this here. There we see only the end. You can imagine that there's a lot of stuff going on. All these are dynamic structures, and people want to understand how these things are connected to each other. And this is the only technology which allows to do this. The first breakthrough in this direction was made 2 years ago by Joerg Lindenau, who imaged the first cubic millimeter of human brain tissue ever. This is now browsable for everybody by this special scanning electron microscope. These cubic micrometer on the synaptic scale can now be imaged or watched by everybody, and the goal is to do this with a mouse brain as the next breakthrough, but nobody knows how to do this because with the current technology it would take 200 years if such a single instrument would scan the slices. So the technology has to be improved, but this is the direction where we want to go. Apart from these large projects, for smaller projects like an organoid, like a couple of cells, there's a variation of this technology available, which does not apply the completely automated sectioning, but kind of semi-automated way by a certain add-on to any kind of ultramicrotome nowadays. And in addition to become, just to go back here, what this thing does, it just lifts out of a smaller central carrier here, which allows also a beginner user to take out section ribbons of not a couple of thousands, but of a few hundred individual sections. And in addition to this, we become faster. What we have done, we have developed a special detector, which is optimized to register the signal in the voltage regime. What is used for this imaging means is 2 kilovolt, and this detector is very sensitive and faster than the generations before, which allows us to image these kinds of serial sections faster, because scanning time is always a limitation. For these projects of 1-2 cells, we talk about scanning times of a day, 2 days, or a weekend. If we talk about the volume of 30 by 30 by 30 micrometers, something like this. So this is doable for normal labs, I would say, couple of hundred sections representing the volume of interest. And we talk about pixel sizes of about 10 nanometers in this case. So these are reasonable projects which can be done by single beam scanning systems in our days. Such a result here, as I said, so we have something, the dimensions here are in the couple of tenths of micrometer in volume. And the second one is, if you now take the ultramicrotome inside the electron microscope, then it looks like this. This is the system what we call the volume. It looks like this, and there should be a movie about the volume. Where is this? No, it's not. Sorry, then I somehow skipped the movie for a certain reason. This was my iPad. What we have here, we have a door mounting, and the door mounting contains an ultramicroscope. This slices the slice from the top of the sample away, and then the image of the plastic block is registered by the electron microscope, and then the next slice is taken off, the next image is taken. In this way, we can also generate volumes, but in a completely automated manner, and you don't have the hassle to prepare it by yourself, because you only put a block which has to be pre-print. You put a block inside the electron microscope. So to say, one of the easiest technology for volume approach in our days, because it's completely automated, and the major application for this is organoids. This is what we say. C as applications. So this is an intestine of chicken, I guess, with different types of cells, which can be also recognized here, and other organoids like cancer cells, tumor spheres. So these are the dimension of cells which has to be analyzed in 3D. We use this type of serial block-based imaging for this, what we call the volume. The last one of these three is the one with the highest resolution, but also the one which is the most expensive equipment, or requires the most expensive equipment. Nevertheless, the results are very beautiful. What we get out of this, this is a marine organism, which was completely scanned by focused ion beam imaging, 3D rendered with our own software, and you see all the internal structures of this, but this is the only result. I would like to show you how you get to this. The general purpose here is that you have not only the beam for imaging on the system, but you also have a focused ion beam which ablates material from the sample. And this is shown in this movie. We have to put a plate of metal on the sample as a guide for the knife with some kind of structures which tell us how far we cut in the sample. The carbon layer which we add to get the contrast. Now this ion beam is in action, takes material of the sample off, we polishing the sample, and then we take an image of this freshly created surface, and then we repeat this process, and in such a way we get information about the volume in here. The advantage of these technologies is very, very high resolution. So what we maintain here, we maintain a Z resolution size thickness in a nanometer range, about 5 to 3 nanometer in this case, and therefore this technology can be used for very high resolution in brain tissue. You see here postsynaptic membranes, vesicles, all the structures what we need to understand for electronics can be easily displayed. Nevertheless, so here volume and pixel size is in the 3 by 3 nanometer regime in this case. Let's skip over this. We can do both imaging with this. The time for such a cell is also in a 20-30 hour for this kind of a volume of a cell. Ablation imaging. But in the end, we have the resolution which is required to see all the structures in here. Here you see volume of this pixel size is in a range of, we can go even smaller, but 7 nanometers here, 30 by 20 by 25 micrometer, but it takes 2 complete days just for scanning. Yes, with this one, if you want to go to bigger samples, then there's a problem in this case, because we have to dig these kind of trenches into the sample surface, and there's also redeposition, and this redeposition of material becomes bigger and bigger as deeper we dig into the sample. But nevertheless, there's technology which is called minimum resonance embedding, where we put the samples on sticks, and this works for insects, for nematodes, for C. elegans, for instance, and then we don't have to cut the material around of these samples. We just have the sample on the surface of the resin, and then we cut only into the sample itself. So this gives better contrast, less redeposition, and allows us to target our imaging throughout the structures of the cells, kind of correlation method. And this has been done by the EMBL in Heidelberg, for instance, at a 3 nanometer regime, 10,000 sections for the complete C. elegans larvae. And these larvae is now one of the first organisms where the complete neural system was reconstructed on the base of these type of images. You see such an example. This is the EM image itself, and this is the reconstruction of the nerve system of this part of the worm. I have one more, and this is the same technology. This focused ion beam milling can also be used to look at non-labeled samples. What you see here, the quality is a little bit less compared to the quality of the images we have seen before. But the special thing here is there is no label on this, it's just a frozen sample. And what the image here is, we can see membranes of the nucleus, of axons, of mitochondria, and so on, just due to the fact that the membranes in the eyes have a different conductivity as the eyes itself, and therefore this is enough to create a contrast here. This somehow gives a link to dynamic imaging, if we start with a light microscope, for instance, under room temperature condition where the sample is alive, and to transfer these samples under cryo conditions into an electron microscope. How does this look like? What we can do is we can have this device which is called the high pressure freezer connected to a light microscope. We have the sample here above a light microscope in a little dish where the sample is still alive under room temperature conditions, and we can image this sample, select a certain time point we're interested in, and then click a button and pull this sample here above the objective lens into the high pressure freezer. What we then get is this process takes about 1.5 seconds. So 1.5 seconds after we've seen the event, we have the sample frozen in ice. And this can be just a sample without any label, but it can also be a fluorescently labeled sample. What we do in the next step is that we take the sample which is now frozen back to the light microscope and image this with a cryo stage under the light microscope, just to confirm the location of the fluorescence inside the sample. And then we take the sample out and transfer this to a special type of electron microscope under cryo conditions and look at the same sample under cryo conditions. Then there's two different options in the cryo electron microscope: either to perform volume imaging in the electron microscope, or you prefer a lamella for TEM for ultra high resolution imaging. It's probably these kind of images. You know, this is a reconstruction of images obtained with the SEM under cryo conditions as well. And this is ultra high resolution molecular image of certain structures in a very thin lamella created here from the ice. The equipment for this workflow look like this. We have a light microscope with a cryo stage on it. We have a shuttle box, and we have an electron microscope which also operates under cryo conditions. This shuttle system provides adapters and holders for the different systems. The sample go into one of these holders, and then according if you go to the light microscope or to the electron microscope, there are different adapters to put these sample on it. First of all, you would go with such a sample in the light microscope under cryo conditions. This is the little this is the cooling stage under light microscope. Afterwards, you transfer the sample into the electron microscope and perform the imaging in the electron microscope. I would like to show one example. This is Nico Sommerdijk from University in No, and he moved from Eindhoven to Nijmegen. Sorry, he moved from Eindhoven to Nijmegen, so he's in Nijmegen now. So what he's doing, he freezes samples with a finder top, performs fluorescent imaging at different channels. In 3D, you see the ice surface as well, the mitochondria and the cells. Then he puts this sample into the electron microscope, overlays what he sees in the electron microscope with the data from the fluorescence microscope, cuts a trench into this, images his collagen layers, which are fluorescently labeled, in the electron microscope, overlays this with the fluorescence data, and in the end he has fluorescence data and electron microscopy data from exactly the same structure. And this can be done up to a resolution of 5 by 5 by 5 nanometer in 3D in the electron microscope. So this is a big advantage of this one, and you can see single collagen fibers here, cross sections, how the orientation looks like. And my last slide is a movie, which is not here. So just thanks to Nico Sommerdijk and his group for this fantastic work what he has done on correlation between living fish scales and frozen ultra-scale microstructures. Thank you very much.
you. I was told by the organizers to speed up here because we're behind schedule, but one urgent question, and then I know that Joerg is anyway present over there. Do we have one? Yeah, we know where to find him. Thank you very much for showing up.